Okay, bueno, we are going to start. I'm going to switch to Spanish. So, bueno, muchas gracias a todos por estar aquí conectados para este seminario del programa de Frontiers in Genomics, que es organizado por la Licenciatura en Ciencias Genómicas, el Centro de Ciencias Genómicas y el Instituto de Biotecnología. Y como saben, los que ya han estado en estos seminarios, hasta el final son las preguntas. Aquí pueden alzar la manita aquí en el chat para que yo les dé les pase el micrófono, para de alguna manera decirlo, para que formulen la pregunta, o la pueden escribir directamente también en el chat. Bueno, habiendo dicho eso, para mí es un gusto presentar a la doctora Catherine McCusker de la Universidad de Massachusetts en Boston. La doctora McCusker hizo su licenciatura en Química y Bioquímica en Bridgewater State University en Massachusetts. Después, en el 2010, completó su doctorado en el programa de Biología Celular y Molecular del Departamento de Veterinaria y Ciencias Animales en el laboratorio del doctor Dominic Alfandari sobre las interacciones moleculares que controlan el desarrollo craneofacial en Genopus levis. Antes de eso, también fue Health Research Technician en the Mass General Hospital en el Departamento de Radiología dentro de la División de Medicina Nuclear, en donde interesantemente realizó investigaciones sobre el desarrollo y pruebas de drogas que son marcadas con radioactividad. Sí, por eso estaba en ese departamento. En el 2009, del 2009 al 2015, realizó un postdoctorado en el grupo del doctor David Gardiner y Susan Bryant, en donde trabajó sobre el papel de la información posicional en la regeneración de los miembros de Ambistoma mexicanum, y esto lo hizo en la Universidad de California en Irvine. Y desde el 2015 es Assistant Professor en la Universidad de Massachusetts en Boston. La doctora McCusker ha organizado conferencias importantes, todas relacionadas en biología del desarrollo, y ha recibido donativos importantes para su trabajo de investigación, que incluyen el NIH, el National Cancer Institute y la American Cancer Society, entre otras instituciones. Y como ven, es una investigadora joven, y a pesar de eso tiene ya reconocimientos importantes que incluyen la Society of Developmental Biology y la presentación de una plática plenaria en el curso de Frontiers in Stem Cells and Regeneration en el Marine Biological Laboratories in Woods Hole. Y el día de hoy nos va a hablar sobre the induction of positional plasticity and the regenerating axolotl limb. So, Catherine, thank you very much to be here with us, and now we are ready to hear your talk.
Okay. Thank you so much, Mario. Thank you for hosting me, and thank you for the nice introduction. Okay. So yesterday I had a wonderful class with the Frontier students, and I wanted to mix things up a little bit because I presented to them another project that we have going on in the lab. So I wanted to keep it fresh for them and present something that was different. Let me close this so I don't have too many things open at once. Okay. All right. So the project that I'm going to bring you guys through today is focusing on a really interesting problem that regarding regeneration. And that is, how does that regenerating limb grow to the correct size that's proportional to the animal that it's growing on? And I wanted to start off by pointing out this student, Dr. Kaylee, who's now Dr. Kaylee Wells. She was a PhD student in my lab who has started her postdoc at Harvard. And she's really responsible for getting this project off the ground. It's a totally new area of research for my lab. Okay. So we study limb regeneration in my lab, and it's not, entirely surprising why we're trying to study how limbs regenerate on the Mexican axolotl. Whoopsie. So limb amputations and limb injuries are a major health disparity, as well as a major cause of changes in lifestyle and quality of life in the patients that suffer from limb loss. in the United States alone, there are over 2 million Americans that suffer from limb loss, and there are a variety of different causes. I think we typically think of a traumatic injury as leading to limb loss, but there are a lot of diseases that are highly prevalent throughout our populations that can also lead to limb loss, including diabetes and cancer and so forth. So the objective of my lab and many labs across the world studying tetrapod limb regeneration is to really understand the basic biology of how these animals can regenerate these complex structures, to eventually, extract those lessons from those systems and apply them to developing regenerative strategies for humans. So the good news is that humans do have a very little bit of regenerative ability in their limbs. So it turns out that we actually can regenerate our digit tips as long as there's some of the nail bed present. And this includes the bone, the muscle, the connective tissue, the skin. And actually, this process of regeneration shares a lot of similarities with the process of limb regeneration in axolotl. So it looks like, many of that aspects of digit tip regeneration are conserved between tetrapods, which is very, very encouraging. But the sad thing is that if we do an amputation just a little bit further down on the hand, so this would be in the P2 element, or obviously further down on the proximal axis of the limb, those amputations result in complete regenerative failure. So the idea here is how to figure out what's different in these types of amputations compared to these types of amputations that lead to regenerative failure, and potentially we could kind of tap into these differences to develop regenerative therapies. So humans have a little bit of regenerative ability, but it is, as you know, very, very limited. But the Mexican axolotl and other Urodale amphibians like newts and salamanders are excellent at regenerating their limbs. And in fact, they're kind of excellent at regenerating lots of different things. And here is just a sampling of the different types of structures that people have studied limb regeneration. And, I'll just point out a couple that are particularly interesting. Is there a question? Oh, I think maybe somebody has their microphone on. Okay, so one of those aspects that I think is really interesting is their ability to regenerate their spinal cord and their vertebrae. This is different from lizard tail regeneration, which some of you might be familiar with. The lizards regenerate more of a biological prosthetic where when they lose their tail, they actually regenerate just kind of a rod of cartilage that's surrounded by connective tissue, and that's surrounded by skin. So it replaces the function and kind of superficially looks similar, but it's actually not replacing exactly what's missing. The axolotl, on the other hand, does replace exactly what's missing. And so understanding how they can regenerate their spinal cord is obviously very, very important for potentially designing therapies for paraplegics. They're also capable of regenerating parts of the brain. And this was known for some time. So this was a paper from 2013 from Malcolm Maiden's lab, where they essentially removed 1/4 of the brain. And not only did the animal live, but it regenerates the missing portion of the brain perfectly. And there was a recent paper that came out also with Malcolm Maiden as one of the PIs. And that's kind of further pushing this area of research. But I would say definitely by far and large, most of the people that are working with this model organism are studying limb regeneration. And that's really what we're going to focus on today. So I'm going to start off with a bird's eye view of limb regeneration. So here's an amputated limb. Oh my goodness, sorry. Here's an amputated limb. And you can see that ball of cells growing at the severed end of the limb. Those are blastema cells. They're essentially de-differentiated adult cells. And those cells are going to act as the progenitors for the regenerating limb and grow and pattern into the missing limb structures. I'm just going to tell you a little bit of basic biology about how regeneration happens, especially during those early stages of blastemal development. So you need to essentially have three basic requirements in the limb environment to be able to establish a blastema, which is that transient regenerative organ. So the first requirement is the formation of a wound epithelium. So what we're looking at right here is kind of a profile through a limb that's been amputated. And the wound epithelium essentially migrates over and completely covers that severed edge of the limb within hours after amputation. And so we essentially have a direct interaction of those keratinocytes with all that underlying mesenchymal tissue, including the bone, the muscle, the connective tissue, the nerves, and so forth. So that's the first requirement of limb regeneration. And the second requirement is that you need that severed end of the nerve to regenerate and innervate the wound epithelium. And there are signaling feedback loops that are happening between the nerve and that wound epithelium that essentially establish a specialized wound epithelium that's very, very similar to a structure that also forms on the developing limb bud in the embryo. And in fact, a lot of these factors that are generated in this specialized signaling center, it's known as the apical epithelial cap, are the same ones that are generated during limb bud development. So there is this kind of conservation of the underlying mechanisms that are playing a role in the activation of the blastema cells. So those factors that are secreted by that apical epithelial cap are absorbed by those underlying mature cells. So these are adult cells that are receiving the signals. And the end result is that these cells start to undergo a process of de-differentiation where they lose their adult characteristics. They start to express unique markers to the blastema. And they also start to proliferate and migrate towards this specialized wound epithelium. So this is how that early blastema forms. And we have a surgery in the lab that was originally designed at Dave Gardner's lab, which is the lab that I did my postdoc in, where if we supply with, or if we provide the limb with these two aspects, that would be a wound epithelium and an innervation, we can actually trick the limb into making a blastema. So here we make just a little wound on the side of the arm, removing a square of skin. And we dissect a nerve and deviate that nerve into the middle of the wound site. And you can see over time that wound site will develop a blastema. But that blastema will actually regress back into the limb and not form any structures unless you supply a third requirement in that environment. So the third requirement is the establishment of positional disparity. And essentially what a positional disparity is when cells from the different limb axes interact with each other in this regeneration competent environment. So for example, if you were to amputate the limb here, cells from the anterior side and the posterior side would migrate into that wound site so that they can accumulate below that apical epithelial cap and interact with each other. And somehow this interaction between cells from the different limb axes is required to draw the blueprint of the missing structure. Okay, so going back to the surgery that I was just introducing to you, if we supply all of these three different aspects by making a wound, deviating a nerve to that wound site, and then grafting tissue from the opposite side of the limb into that wound site, these will result in the formation of ectopic limb structures. And here you can see one example of an ectopic limb that was formed on an animal from one of our studies. So a couple things I want you to take away from this surgery that I just introduced to you. So one thing that has been very, very useful in my lab as well as in some other labs is that this surgery breaks regeneration into those three different major components of the regenerative process. And so it allows us to dissect those things out individually and figure out what the molecular mechanisms are and the cellular contributions and so forth. So it's really helpful in that way. The other thing that is important, at least for some of the studies that I'm going to show you today, is simply making a wound site and deviating a nerve to that wound site. And you don't need to graft this tissue, but just doing those two things alone will make a regeneration permissive environment. And that is something that we can really tightly control and leverage for other situations. And that will make more sense when you see some of the other experiments that I'm going to show you today. Okay. So this is a time series of limb regeneration on an animal, and those stages of blastemal development that I just talked you through are occurring during these days, like, you know, up to about day 31 or so. It depends on the size of the animal. It takes more time in large animals. But one thing that you probably notice is that there are pictures of limbs well after this day 31. And the reason why those are there is because if you compare these regenerating limbs to the limbs on the other side of the animal that had not been amputated, you can see that they're really small in size, and they grow fast until they essentially catch up with the limb size on the other side of the animal and are proportionally appropriate. And then they slow their growth so that they grow at the same pace as the rest of the animal. And this was absolutely fascinating to us. And we decided to focus on these stages of growth and understand what the underlying mechanisms are that regulate this process. We also named this the tiny limb stage of regeneration because the limb is tinier than the unamputated limb. Okay, I also wanted to take a step back and kind of, even though this is a very basic biology question that we're asking, if you think about it, it's really, really important for us to understand the answer of this question for human therapies. And if you think about it, the size of the embryonic limb when it's developing is much, much smaller than the limb that needs to regenerate, right? Because we're planning on regenerating limbs on adults that need it. And this seems like kind of an insurmountable goal, right? Like here, the size of the embryo at this point is about 10 millimeters. And the size of this is much more than 10 millimeters. But I would say that the axolotl gives us a little hope in this regard. So the axolotl is an indefinitely growing species, which means that they continue to grow throughout their entire life. And yet they're capable of regenerating a proportionally appropriate limb throughout their entire life. And so I think that this tells us that we shouldn't be so concerned about size because there are animals that can deal with this issue of size. So when we first jumped into trying to figure out the answer of how growth and size is regulated in the axolotl, we dove into the literature and there was really almost nothing out there. And so we started off the study basically just by characterizing growth. And so we amputated limbs off of lots of animals, and we measured the size over time. And so here's what the limb length over body length ratio looks like. And we did that because it was a way to kind of measure proportionality, because there is a little bit of variance in the body length of the animal. And this is what that ratio looks like during those blastemal stages of development. And then at this point, which is just before the 40-day mark, we have a completely regenerated limb that's very small in size. So that's the initiation of that early tiny limb stage. The early tiny limb stage lasts for a few weeks, and then its growth rate is actually quite similar to the growth rate in the blastema. So the change in proportionality is quite rapid. And then over a period of about a week, the growth rate slows down substantially. And although it's growing faster than the uninjured limb, which proportionally stays pretty much the same throughout the entire experiment, it's starting to slow down, and it just kind of gradually slows down until there's no significant difference in the growth rate or the size of the regenerated limb compared to the uninjured limb. And this second stage of growth that we're looking at, we've named the tiny limb stage, or the late tiny limb stage. So we have the early and the late tiny limb that are basically delineated by the switch in the growth rate. And here are pictures of the early and the late tiny limb stage. Okay. Another thing that we looked at while we were characterizing these different phases of growth was how long the regenerating limb was in these different stages, depending on the size of the animal when it was amputated. And so we looked at animals that were 4 centimeters from the tip of their nose to the tip of their tail, 10 centimeters and 20 centimeters. And not entirely surprising, the amount of time that it takes to complete regeneration is much, much longer in the 20 centimeter animals. And this is something that kind of everybody in the regeneration field knows. It takes large animals longer to regenerate. But the thing that I think is most interesting to me about this data is that the pre-tiny limb stage, so these would be the blastemal stages, there actually isn't that much difference in the amount of time that the animals of different sizes are in that stage. It's really the tiny limb stages that are more dramatically impacted in these different sized animals. And that's probably because the limb that's regenerating on the 20 centimeter animal is much bigger, right? And so there's more catching up than it needs to do to be proportionally appropriate. Another thing that we looked at when we were characterizing growth in the regenerating limb was what types of cellular contributions contribute to this growth. And my PhD student, Kaylee, looked at a couple of different things that have been, well established to contribute to growth. She looked at things that could regulate the overall number of cells. So she looked for whether there was an increase in cell proliferation or a decrease in apoptosis. She also looked for changes in cell size. So do the cells get bigger in size during those tiny limb stages of growth? And she also looked for the abundance of extracellular matrix deposition and whether there was an increase in this deposition over time. She was also very thorough, and she did this analysis on these different stages. So she looked at unamputated limbs, early and late tiny limbs, as well as fully regenerated limbs. And she looked at the different tissues that compose those different, staged limbs. So she kind of grossly grouped the tissues into epidermis, the skeletal elements, and then the soft tissue, which essentially was just kind of everything in between the epidermis and the skeletal elements, and looked at all of those different factors that I just told you about. So in all of the tissues that Kaylee looked at, she saw a significant increase in cell proliferation during the tiny limb stages. And by the fully regenerated limb stage, so to speak, things had gone back to the unamputated limb abundances. She also looked at cell death, which decreases during those tiny limb stages and then increases back to unamputated limb levels in the fully regenerated limb. She interestingly also saw changes in cell size. So the cells in the early tiny limb are smaller than the mature cells that are in the unamputated limb. And then as those limbs continue to develop through those tiny limb stages, they increase in size. So cell size could be, or changes in cell size could be another contributor to growth. But she did not see any significant changes with the abundance of extracellular matrix in the tissues. So we've stopped essentially looking at that in our future studies. So now that we had characterized, what was happening during these stages of growth, we really wanted to understand a little bit more about how this process of growth and size regulation is regulated. And again, there was really not much out there in the literature to give us inspiration, but we did find some things, and so we found that there were different treatments that were shown to have impacted the size of the regenerating limb, either in the axolotl or in the newt system. And we saw that pharmaceutical inhibition of FGF signaling, BMP signaling, or TGF-beta signaling all resulted in the formation of smaller limbs. These studies didn't focus on size. They were doing studies that were focused on different questions, but they did observe smaller limbs when they did that treatment. And then another treatment that has kind of an opposite effect is the treatment of the regenerate with exogenous retinoic acid. And exogenous retinoic acid does something else. It reprograms the positional information in the regenerating limb, but that reprogramming results in the elongation of the skeletal elements, and at a high abundance can actually cause a duplication of the proximal distal structure. So retinoic acid has the opposite effect. It can make limbs bigger than they should be to be proportionally appropriate. The cool thing about all of these things is that these things are things that are normally present in the regenerating environment and are all downstream of signals that are coming from the nerve. So even more in line with this theme of nerve signaling and how it could be regulating size are some studies that have come out in humans, as well as some other studies in other model organisms, such as the axolotl, where the overall abundance of nerve signaling correlates with the size of the limb structures. So what we're looking at right here is an X-ray of a toddler hand. And this toddler has a tumor called neurofibroma on the nerves that are feeding into this digit. And as you can see, the digit, the actual structures that are there look really good, right? But they're very, very large in proportion compared to the other digits. And these types of tumors result essentially in increased neural signaling. So here we have increased neural signaling and an increase in size of the limb structures on humans. And then if we look at this example in the axolotl, essentially, one of our collaborators, Jessica Whited, her lab had generated these animals that had miniaturized limbs. So essentially, they continuously amputated the limb bud starting during embryogenesis, and did that for about a year. And when they stopped amputating the limb bud and allowed the animal to complete regeneration, they regenerated a limb, or I should say generate, because they never actually had a limb to start with. But the limb that they formed was small proportionally to the rest of the animal. And when they look inside of those limbs, and here's essentially a tissue section of a wild type limb that had never been amputated and one of these miniature limbs, and in green, that's a stain for the neurons. You can see that there is a significant decrease in the abundance of neurons that are present in those miniaturized limbs, and they quantified this as well. So here we have a correlation with decreased nerve presence and decreased size of the limb structure. So we were really interested in these correlations, and we wanted to see whether we had a similar correlation in the tiny limb environment. And so essentially we just did a lot of histology sections of the tiny limb at these different stages and compared them to the abundance of innervation in the regenerated limb, as well as the unamputated limb. And in green, again, that's staining for the neurons. And when we quantify the relative innervation, that is the abundance of neurons relative to the area of the limb that they're present in, we see that there's a huge significant increase in the relative innervation of the early tiny limb that gradually decreases as the limb progresses through those later stages of regeneration and goes back to normal in the fully regenerated limb. So here in our tiny limb regenerating environment, we also see this correlation where the highest relative innervation correlates with the stages of growth that are most rapid during regeneration. Okay, so we next wanted to test whether the presence of these limb nerves were required for regenerative growth. And Kaylee, my student, was very thorough in this analysis. She wanted to see if there could be also a dose response, if we even saw a response. And so she severed 1, 2, or all 3 of the nerve bundles coming from the spinal cord leading to the limb, and looked to see the impact on the overall growth of this entire limb, as well as those different aspects that contribute to growth that I already told you about. And to summarize this briefly, she saw that only with a full denervation, she saw a significant reduction of the overall growth of the limb. This is only 4 days after severing the limb. So I was actually really surprised that she saw a significant decrease in the growth rate even after 4 days. And then when she performed her histological analysis, she also saw a significant decrease in cell proliferation, a significant increase in cell death, but not really any significant change in the cell size in the different tissues she looked at. The exception here would be muscle. So probably what's happening, because we see a decrease in the overall size of the muscle tissue after denervation, is that maybe the muscle is atrophying. We haven't assayed for that, but that could be one of the reasons why we see a decrease. So together, this indicates to us that the limb nerves are required for regenerative growth, and we can see the impact of losing nerve signaling within 4 days after removing the nerve signals. So the next obvious question was whether nerve signaling actually plays some sort of instructive role in regulating the overall regenerate size. So to start to tackle this question, we took advantage of the really cool biology of the axolotl, and that is that we can regulate the growth of the animal by growing them at different temperatures. And so these 2 animals are clutch mates. They were born at the same time by the same mother. The difference is that this animal was housed at 4 degrees for 3 months, and this animal was housed at 22 degrees Celsius for 3 months. So there's a huge difference. And when we quantify the limb length and the body length, the animals housed at 22 degrees Celsius are much larger in size than the small animals that you can see here in this picture. And another thing that we looked at was the size of the nerves that were present in these differently sized animals. And we performed a histological analysis looking at the nerve bundles that are coming from the spinal cord that are leading to the limb. And there is a significant increase in the size of those nerve bundles in these large these large animals compared to the small animals. Okay, so we kind of leveraged these differences in size as well as nerve abundance in these differently sized animals to get at this question. And what we did was a series of grafting manipulations where we amputated GFP positive large donors and small donors to generate blastemas. And then we grafted those blastemas to regeneration permissive environments, like the ones that I told you about earlier in the talk, that's made by making a wound site and deviating a nerve to that wound site on either a large host or a small host. And you might notice that the small host, that the nerve is actually deviated to a wound on the flank of the animal. And the reason for that is more of a technical reason. So it's because grafting a large blastema onto this tiny little limb was not possible. The blastema is so much larger than the limb that we couldn't make that happen. So one way that we could make that happen is by grafting the large and the small over to a flank innervation on the small animals. And here you can see that growth of this grafted structure is completely supported in this regenerative environment. And essentially what we did was we measured the overall size of this regenerated structure in all of these different manipulations. Okay, so what did we see? So when we made either large grafts or small grafts to a large host or small host, at the day that we did it, there was a significant difference in the size of the graft, not surprisingly, right? And then we allowed the grafted tissue to complete regeneration, and we determined that they had completed regenerating when the growth rate of that regenerated structure matched the growth rate of the limb that they had been grafted to. So it was isometrically growing with the rest of the animal. And once they reached that point, we saw a couple of really interesting things. So the first was that the size of the regenerate, regardless of whether it was a graft from a large host or a graft from the small host, was not significantly different when you compare between each sample group. So if you look at the large host, there was no significant difference between the regenerate size. And if you look at the small host, there was no significant difference between regenerative size. But when you compare the size of the regenerates that form on the large hosts versus a small host, there is a very large significant difference. So the regenerates that form on the small host are significantly smaller than those that grow on the large hosts. And so this suggests that the limb environment itself, the host environment with these different size nerve bundles are likely playing a role in this overall difference in size of the regenerates, regardless of the animal size that those grafted tissues came from. And just to be kind of safe about this, we made measurements on the donor animals where we had harvested those blastemas to graft to make sure that by the end of the study that there actually was still a significant difference in the size of those regenerates. And in fact, there was. So that significant difference was maintained on the donor animals at 145 days post-amputation. Okay. But if you think about it, even though there were very big differences in the size of the nerves that were on that host environment, either the large host or the small host, there are probably lots of other things that are present or differentially present in the large and the small host environments that could also be contributing to differences in growth. And this was something that we were really worried about. So we actually developed an entirely new assay to tackle this question, where we could separate the source of the neurons or the dorsal root ganglia with the source of, or the host essentially. So we called this the neural modified accessory limb model. And in the NMALM, we have two donors, one donor of a dorsal root ganglia and one donor of a blastema. And then we have a host where we essentially just make a wound site on the host and we implant the dorsal root ganglia from the donor. We have it a GFP dorsal root ganglia so that we can make sure that it survives and is innervating. And here you can see the innervation from that implanted dorsal root ganglia. And then on top of that, we kind of plop that blastema. We harvest the blastema and put that on top. So here's kind of the surgical manipulation that we're doing. And amazingly, this dorsal root ganglia essentially behaves in a very similar way as what it would had it remained intact, and that it can completely support the regenerative process. And as you can see here, it's the regenerating limb is highly innervated. So we leverage this assay to answer two questions. The first question is whether there were non-neural extrinsic signals that also could regulate growth in the regenerate. So in this case, we used the NMALM, but we had two differently sized hosts. We had a large host and a small host. The dorsal root ganglia and the blastemas came from the same size host. So these were 7-centimeter hosts. And we grafted these both over to these differently sized host animals. And the prediction was, if non-neural extrinsic factors are playing a role in the growth of the regenerated limb, then we should see a differently sized regenerate on differently sized hosts when the abundance of innervation stays the same. But if no non-neural extrinsic factors are making a major contribution to growth, then we shouldn't see significant differences depending on the host. And I should say that before we did this study, I really had no idea what was going to happen. But I kind of imagined that maybe something like this would happen, that maybe there was some sort of contribution from these non-neural extrinsic factors, but that's not what we saw. We actually saw the second outcome here, where the regenerating limb was the same size in the differently sized host after regeneration had completed. And just as a control, we also measured the length of the limbs on the control animals. So this would be the size of the host limbs, and those remain significantly different throughout the entire experiment. So this indicates that the nerve is playing really the key role in regulating growth and size of the regenerating limb. The second question that we used this NMALM assay to do was to figure out whether those neurons were providing the instruction for growth autonomously, or whether they need to be connected to the central nervous system to regulate this process. So in this case, we kept the size of the host the same. But the dorsal root ganglia that we harvested were from different size donors. So here's a dorsal root ganglia from a large donor. Here's a dorsal root ganglia from a small donor. And we took those along with blastemas from 7-centimeter animals, all the same size, and grafted them over to that host animal. And in this case, the prediction is that if there is a different size regenerate that forms over these differently sized dorsal root ganglia, then that would indicate that the dorsal root ganglia is kind of acting autonomously. Potentially, it just has to do with nerve abundance. However, if there is no significant difference in size, regardless of the size of the dorsal root ganglia that's been implanted, then this would indicate that the dorsal root ganglia is actually acting somehow as a conduit, that there's a central nervous system connection that's required for them to do their job appropriately. And again, in this scenario, I totally admit to thinking that this is probably going to be the outcome here, where the dorsal, it's all about abundance of neural signaling, and we should see significant differences in the size of the regenerates on the different size dorsal root ganglia. And again, I was wrong, but this is obviously very exciting still. So essentially, we found that there was no significant difference in the size of the regenerate that forms on the small DRG versus the large DRG. And I was a little worried after seeing this. So I had Kaylee go in and harvest these limbs and quantify the abundance of innervation, because I was like, maybe the abundance of innervation is not different in these ectopic limbs that grew over these different sized dorsal root ganglia. But lo and behold, lo and behold, there was a significant difference. So there was significantly more innervation in the regenerates that grew over the large donor, but still this increased innervation was not enough to increase the overall growth or size of the regenerated limb on that dorsal root ganglia. So this is exciting because it indicates that we need a connection to the central nervous system to regulate the size of the regenerate. Let me keep an eye on the clock here. Okay. So this put us, that's pretty much most of the paper that we had published earlier this year in eLife. And we still have been able to kind of target some lingering questions, of which there are many, related to how growth is regulated. So I would say, the three major questions that we're going after is figuring out what that upstream regulation is, like where is the source of instruction that tells the dorsal ganglia how much signal to give. We're also interested in figuring out what are those signals that are coming from the neurons that promote growth in the regenerating structure. And last, we're also interested in figuring out what is the response of the tissues, and whether there are specific tissues that are really responding to those signals, and how they're doing that. So I'm going to focus on some of the studies that we've done to figure out what are those signals that are coming from the neurons. And nobody had really looked at neural signaling during these later stages of regeneration. So to start this study, we took some inspiration from people that had studied neural signaling during those blastemal stages of regeneration. And people have been looking at this for a long time because it's been known since, I guess, the 40s, that neural signaling was essential for the formation of the blastema and the growth of the blastema. So there had been a lot of data on that. And more recently, people had tried to start to figure out what are the actual molecules that are generated by the neurons that support these early stages of regeneration. So here's just a couple of really high profile papers that focused on this question. Here's a paper out from Akira Sato's lab in Japan, where they found that BMP and FGF signaling inputs can take the place of the nerve, so they can support these early stages of regeneration with beads soaked in BMPs and FGFs. This is a very well-known study that came from Jeremy Brox's lab, where they found a molecule known as anterior gradient. The study was done in newts, so NAG. Also can take the place of nerve signaling. And then this is a study that came out from James Monahan's lab up here in Boston, where they found that neuroregulin 1 also can play the role of nerve signaling. So what we've done is we started just looking at the expression of these different factors in the dorsal ganglia in these differently staged regenerates, and compared them to the expression profiles in unamputated and regenerating limbs. And essentially across the board, all the factors that had been previously identified as these nerve-dependent factors are significantly more expressed in the early tiny limb when growth is most rapid, and kind of change in expression depending on the factor that you're looking at over time. So that was interesting. It told us that maybe we were on the right trail. We also looked at the expression of these factors in the regenerating tissue. And we found that a lot of them were also increased in expression in the regenerating tissue, with the exception of neuroregulin 1. And I have to say it's not entirely surprising, because neuroregulin 1 is really something that's isolated to the neurons, whereas these other factors do play key roles in kind of the development of the tissue and the general function of the tissue. So that wasn't entirely surprising. But again, we saw these trends where there was a large amount of BMP and FGFs that were present during those early tiny limb stages. So we next wanted to see if some of these things that were expressed in during the tiny limb stages were whether their expression was dependent on the presence of nerves. So again, we looked at the expression of these different factors, but we compared their expression in innervated, where the nerves were intact, versus denervated regenerating limb tissue. And these were at the early tiny limb stage. And we found that there was a significant reduction of these two BMPs. GDF5 is another BMP. But very interestingly, we saw that there was actually a significant increase in FGF2. And this was particularly interesting to us because we were familiar with the growth plate research that had been conducted in other tetrapods, which show that there is this really kind of tight relationship, complementary relationship between BMP signaling and FGF signaling in, oh my goodness, sorry, and the regulation of these different processes in the growth plate. So by far and large, BMP signaling promotes the growth of the growth plate. And it does this by promoting proliferation in the resting zone, in the proliferating zone, as well as maturation in the hypertrophic zone and the ossification zone in the long bones. BMP signaling is also known to inhibit FGF signaling. And FGF signaling, in fact, has an inhibitory role on these different processes. So increased FGF signaling inhibits long bone growth, whereas increased BMP signaling promotes long bone growth. So we decided to go in and just measure whether innervation was directly impacting the growth of the long bones. And we did this at 2 different time points following denervation and measured the total length of the skeletal elements in the regions that we knew were regenerated. We did the amputations in the mid-humerus, so we didn't want to measure this element because we couldn't control for exactly where that amputation plane is. And even at 4 days post denervation, we could see a significant decrease in the length of the skeletal elements on those denervated animals. And this gets even more significant by 8 days. So we felt like we were kind of on the right track. So the next thing that we wanted to do, given that BMP signaling seemed to be nerve dependent, and BMP signaling had previously been shown to inhibit FGF signaling, we wanted to see if BMP signaling itself was required for tiny limb growth. So we made use of a pharmacological inhibitor for BMP signaling. And we treated early tiny limbs with this inhibitor and measured them prior to the treatment or after treatment to see the overall growth. And we also did a molecular analysis looking for the expression of different genes that we were interested in looking at. So we found that there was a significant reduction in the overall growth rate of the limb in the treated animals, in the BMP inhibited animals. And interestingly, there was no difference in the growth rate of the body length of the animals. So this change really was focused on that regenerating structure. We also looked at Mfx2 expression. Mfx2 is directly downstream of BMP signaling, and we saw a significant reduction of Mfx2 kind of as a control to show us that the BMP inhibitor was working on BMP signaling in the axolotl model. And then we looked at the expression of BMP2, FGF2, and FGF8. So we found that there was a significant reduction of BMP2 expression in the inhibitor-treated limbs and a significant increase in FGF2 expression. And this profile is really interesting because it matches that we saw in the denervated limbs. On the other hand, though, we didn't see a shift in FGF8 expression where we did see that in denervated limbs. So BMP is probably one of the major players in regulating kind of the gene expression patterns that could be playing a role in growth, but it doesn't regulate all of them. So there are more question marks that we now need to answer. But essentially, we found that BMP signaling is playing a role in the suppression of FGFs. We also know that nerves are doing that. We don't know if it's all through BMPs or whether there are other factors that are suppressing nerve signaling, but there are a lot of dotted lines here. And so we hope to tackle at least some of these dotted lines to figure out whether BMPs and FGFs that are supplied by the nerve or that are present in the regenerating limb are playing a role in regulating these different aspects of growth plate function. Oh, and we are also interested in looking at the upstream signals that are regulating the neural function in this process. So this is kind of where we're going from the future. We're essentially going to focus on those upstream signals and to try to identify the source of size instruction. We're also still trying to piece together the signals that are coming from the nerves and how those signals are received from the regenerating tissue. And once we really get a grasp of the molecular biology of all of this, eventually we want to be able to answer this really big question, which is how is growth slowed down as regeneration completes? So this project is really in the early days, and we have a lot more to learn about this question. I have a small army of people that were working on this. Many of them actually were undergraduates. So Kaylee, my PhD student, was the major player, but there were also a lot of undergraduates that played a role in generating this data. We have funding through the National Institutes of Health, and I will take any questions that you guys have.
Okay. Thank you very much, Catherine. Very exciting results, very interesting. So now the presentation is open for questions. There's a question from Luis Covarrubias. Go ahead, Luis.
Oh, I think you need to unmute yourself.
Thank you for your talk.
We cannot hear you well, Luis.
I have 3 questions. One is regarding the innervation. Is there any molecule identified that attracts nerves to the growing?
That attracts nerves to the what? You cut out at the end.
We cannot hear you, Luis. No te escuchamos. You can write your question.
I think maybe I'm going to take a guess at what Luis was asking, and that is whether there's anything present maybe in the regenerating environment that is attracting the neurons. And if that is your question, the answer is yes. There is absolutely feedback between the blastema and that wound epithelium that are attracting the neurons to innervate that tissue. Is that getting at the heart of the question you were asking?
Yes. I was thinking more in semaphorins or things like that are known to promote growth of nerves in the nervous system. I was thinking that there was something like that in the limb.
Probably. I'm not aware of anyone that's actually directly looked at semaphorins, but we had done an experiment in my lab where we had so we were trying to see if we could make extra big limbs by implanting multiple dorsal root ganglia nerve bundles into the limb. And we found that there was no significant difference when we did that. But when we actually looked at the abundance of innervation inside the limb, there was no significant difference, even in the limbs where we had implanted a lot of the dorsal root ganglia. So that to me says that there's probably something in there that's regulating nerve abundance in the tissue, and it's probably semaphorin based, but we have no evidence of that. So yeah.
Okay. My other question is regarding the active molecule that promotes growth. What's apparent from your graph that actually is rather than promoting proliferation, is promoting survival, because you have a lot of cell death when it's innervated. Then again, I'm going to be looking for the different possible molecules. I mean, have you tried to identify those that promote survival rather than proliferation, that help you to identify what is happening?
Yeah, well, some of these factors actually do both. So BMPs can do both. They can act as a mitogen and act as kind of a protectant in terms of apoptosis, like a survival mechanism. So it could be molecules that are doing kind of one and the same thing. But what I would really love to do is just to do like a global expressional analysis. So because we have a very limited view of what's going on, kind of based on the previous literature of different factors to look at. And so I would really like to get, do some RNA sequence analysis or something like that on all of these different tissues over time, so that we have the, just a broader view of what's happening, and we can have more context to the, molecular changes that are happening.
And my last question is regarding the size of the limb. What limits the size? And you mentioned something about that, but I'm thinking, for instance, talking about innervation, is the process of denervation when the growth stops, or nothing to do with the innervation? And my other question, just probably in a different aspect of the same thing, is that someone has studied the hippo pathway, because this hippo pathway has been proposed to the size of organs. I don't know if the same happening in the limb.
Yeah. So I guess I'll answer the second question first. So we have looked at hippo signaling. And people have looked at the early stages during blastemal development, and there definitely are, like, YAP is located to the nucleus during those stages where there's a lot of proliferation. We have not seen significant differences in the expression of the same factors like fat and docious that are responsible for providing some of these signals during the stages of these later stages of regeneration. So maybe still, we haven't done a comprehensive analysis on that, but right now it doesn't look like they're playing a major role in that. And then remind me of your first question again.
In the denervation, if the denervation is associated with the stopping the growth phase of the, this is denervation or nothing to do with innervation. This is a different.
Okay. Yeah. So this is something that it's kept me up late at night, many nights, thinking about, like, how does this whole mechanism slow down? And I think we can take a little bit of inspiration from the characterization studies that we did in the very beginning of all of this, where we just looked at the relative abundance of innervation. And that relative abundance is highest during the early tiny limb stage, and then gradually decreases back to the normal relative abundance as an unamputated limb. So relative innervation, the amount of nerves that are present in a given amount of tissue may have something to do with that. It's almost like perhaps the signal that's coming from the nerves is diluted as the tissue grows, which I think is a very smart feedback mechanism. But we also, as I just showed you, saw very dynamic changes in the expression of different growth factors in the nerves, depending on the stage of the regenerate that they're connected to. So that indicates that somehow the nerve is sensing what stage limb regenerate is connected to and is changing its expression pattern accordingly. So that would indicate maybe there's some type of feedback or something like that between the regenerating limb tissue and the nerves. So I would say there's probably multiple things that are playing a role, but that is a question that I'm really excited to eventually answer down the road.
Thank you very much.
Thank you.
Okay, next question, Joseph Dubrovsky.
Yes, hello. Thank you very much. That's exciting talk and exciting work. I want to ask you about early stage of blastema development, and specifically before digit formation. And my question is in relation to polarity and proliferation. So how cells know where to grow, how the blastema is the directed growth is controlled. It apparently means that in terms of proliferation, there should be the organizer or something like that behaves to maintain it. It is somewhat similar to root meristem, for example. There is a organizer and proliferating cells. So how this system is organized in this context?
Yeah, so in this case, that specialized signaling center, the apical epithelial cap, really at least organizes cell proliferation. And that partially is because a lot of the factors that it's secreting act as chemoattractants, and they also act as mitogens. So they're attracting cells from the stump as well as promoting their proliferation. Now, as the blastema continues to grow, because the cells are proliferating, the cells that are furthest away from that apical epithelial cap start to initiate redifferentiation. So they've specified now and they're redifferentiating into the missing limb tissues, but the cells that are at the very tip, which are still close in proximity to that signaling center, continue to proliferate and to grow kind of outwardly. And then eventually that stops when essentially the completed pattern has been made.
All right, but how do they know where to grow? It could be callus-like growth. Let's say, for example, they can grow radially, and actually they grow radially partially because you say that the length of the limb in the early stage is shorter, but it's thicker. And at least from your pictures, I saw that. Mm-hmm. So that means there is still directional growth maintained. Mm-hmm. And so how this is regulated?
Yeah, nobody I think has really looked at that question. A friend of mine who studies limb development in Canada, I think is starting to tackle that question from a very biophysical perspective in terms of, biological forces that are coming in from different angles because of the growth, because of the tension of the mature tissue compared to this growing tissue that's very loosely organized and so forth. So, we definitely don't have an answer to that question in the regeneration field, but I think hopefully my friend Sivan will be able to figure that out in developing limbs, and we can maybe apply some of that back to the regenerating system.
Okay,
yeah.
And also, question. Right, thank you. And also just one comment and maybe suggestion. In plants, we use not rate of growth, but relative rate of growth. Maybe this parameter will help you to figure out real differences between rates of growth when they are present.
Okay.
That's simply a suggestion.
Thank you. Yeah.
Okay. Thank you very much.
You're welcome. Thank you.
So the next question, Gustavo Pedraza.
Hi, thank you very much for your very nice presentation, very exciting data. My questions were related to the ones that Luis did already. So just to make sure that I got it properly, the levels of FGF2 come up once the limb reached its final size after regeneration?
No, so the FGF expression is really dynamic. So it's actually highest in the early tiny limb and then kind of gradually decreases. And so initially we were thinking, because FGF is a mitogen, we were thinking, maybe that's one of the things that promotes growth. But then when we denervated the limbs and looked at FGF expression, we found that FGF expression actually increased even more. And so we were like really confused by that. And then I cracked open my basic developmental biology book and I was like, wait a minute, FGF actually plays an important role in inhibiting growth of the growth plates. And so then it started making a little bit more sense. And that's why we kind of developed this model of, feedback between BMP and FGF signaling in the growth plates. But I mean, we still have no idea really what's going on.
Yeah, but let's say that at the beginning, when everything is already settled, there is no more proliferation. It's FGF2 up one more time, as making sure that there is no more proliferation and keeping the actual size of the organ?
So we've only looked globally at the entire tissue. What I would really like to, and the answer to that question is when we look globally, no, we see FGF go back to the same level as the uninjured tissue. But I would really like to look at just the skeletal elements and the growth plates and see whether we do see a little bit of dynamics there where like maybe FGF is higher at these early stages, but then as things slow down, or lower, I should say, at the early stages, that things slow down, it kind of peaks out and then, and then I don't know what happens after that. Maybe it goes back down. But that would be my prediction as well.
Okay, thank you very much.
Yeah.
Okay, I don't know if there are questions in YouTube.
I don't see. Oh, I'm looking at the chat. I don't see anything in the
chat.
No,
they are not in the chat, but I don't know.
Yeah.
Sometimes, well, if they are not, so I have two questions, maybe kind of weird questions.
I love weird questions.
I don't know if they are kind of fantasies, but It could be possible instead to use nerves from the axolotl, to put nerves from other organisms. For instance, another vertebrate that do not regenerate, for instance, Xenopods nerves, and see what happened, just to see if there's something intrinsic in the neurons of the axolotl, of the organisms that regenerate, or is common in other nerves, in other organisms. This is, well, go ahead.
Well, we haven't done that, but that's a really that's a fun experiment that we could potentially do. I just need to find a frog person, but yeah.
Okay, and that could be possible to do. I mean,
yeah, absolutely.
Okay,
yeah.
So and the other one is also kind of crazy. I don't know. What's happening? You can take a blastema and put it in a petri dish, and then put the nerves there and in some kind of media, and to see if there's some growing or kind of regeneration, that something like try to create a kind of organoid in vitro, based on blastema cells and nerves. I don't know.
Yeah. When I was a postdoc, I was mentoring a PhD student in the lab that was trying to do just that, and he could maintain the blastema for about 2 weeks and have minimal senescence and still maintain proliferation. And then after that point, things started to differentiate or die. So it works up to a point. It works for a couple of weeks, but then after that point, there are lots of variables there, right? Because it's a lot of tissue. So part of it could be that, there's not enough nutrients like penetrating the blastema, which is fairly big in size. But yeah, people have been playing around with that. Like, wouldn't it be cool if we could grow a limb in tissue culture? The other thing that's kind of tricky, though, is that it takes a really long time to make a limb. So, these are experiments that take months and months to do. So
yeah,
it is challenging.
But at least to see some growing and differentiation, and it could be possible. Yeah. Comparing with the normal tissue in the organisms. I don't know, making expression analysis, just kind of things like that. But okay, it's fine. Okay. So, well, I don't know. There are no more questions.
I want to do another question, Mario. Just regarding your comment, I don't know if, for instance, in the case of innervation is a property that you have studied in the axolotl, but I believe, I'm not sure, that innervation is also required in mammals to the repair of the epidermis. Then it appears that innervation is not a factor that limits mammals for regeneration. I mean, there are additional. I don't know if I am right, but I am saying, is it correct to say that growth promoted by innervation is not what makes the difference between axolotls and mammals?
Well, I would say, certainly in the context of tissue homeostasis, nerve seems to play a conserved role in essentially maintaining the size and the growth of the tissue. We don't have much comparison when it comes to blastemal development, right? Because mammals don't really make blastemas when you have a proximal amputation. But I would venture to say that probably innervation, or how the nerves are reacting to injury, is quite similar between the two. But I think, nobody's actually really directly tried to answer that question. So it would be really interesting to do comparative analyses at the same time between the axolotl and a mammalian system to kind of get at the heart of that. Yeah.
Okay. Well, I think that's it. Well, Catherine, thank you very much. It was very, very, very fascinating results. Very interesting.
Thank you.
And I hope next time it will be in person. Next time that you come and present your work in here in the program of Frontiers in Genomics. And thank you to everyone. So we'll see you next week, guys. The usual guy that always assists to this meeting.
Thanks, everyone. It was fun.
Okay.
Bye-bye.
And as soon as I can, I will send you the bill.
I'll frame it.
Yeah. I was thinking that's a lot. Okay.
Thank you, Mario. Bye-bye.
Bye-bye.